Okay, welcome back. So, good afternoon. My name is Yansheng Liu. I'm a postdoc in episode group for more than already 4 years. Today, I would be happy to discuss with you some knowledge about targeted method of data extraction in DIA and SWATH. My aim is to let everybody understand as much as possible. So I will be very simple in the beginning, and then we can see how we can where we can go. So about besides the targeted method, I would also discuss about the untargeted methods that are recently being developed in other labs. So hopefully we have a better conception about what is the difference between targeted and untargeted method. So here is the outline of the lecture. First, I would present you some general strategies of data analysis of DRA and SWATH. So why do we need to develop the specific strategy for DIA data? That is certainly because of the complexity. And then I would present you some ideas clarifying the difference between targeted and untargeted data analysis. Then in the most of the time, I would talk about the targeted data analysis details exampled by open source, and then the untargeted data analysis exampled by DIA Empire. Finally, with some closing remarks, we will make a summary. So this outline slide will show several times to position ourselves during the lecture. First, the general strategy for data analysis. Here, I review these three core techniques again involved in targeted proteomics: SRM, SWATH, and PRM, and their variant method. So just want to position ourselves in this technical point of view that we are discussing here, the data analysis for SWATH and the DIA. So why do we need to develop specific strategies for data analysis? Here you can see again this simplified version of SWATH acquisition, where we have the MS1 scan and the 32 MS2 scans following that. And this kind of circle will be repeated thousands of times during the LC gradient. And in each window, we open 25 Dalton whites in the conventional SWATH method. So that's why the MS2 spectra is very complicated. So if you think about this repeatedness, finally what you will get is a MS1 map acquired over the LC gradient, and then 32 MS2 maps acquired over this LC gradient. So it is a much more complicated data set compared to the traditional shotgun proteomics, which certainly requires more complicated data analysis method. So if we face something that is unknown in a scientific scenario, how to really learn from it, how to analyze the data? Normally, there are 3 steps. We should first extract some information, and then we should compare this information extracted into something known, which is a benchmark. And then we score this process of comparison. So this concept certainly also applies in the data analysis for DIA or for SWATH. Here we have MS1 and MS2 maps, so they have some internal relationship. One thing that we can try to use is the spectral library generated from shotgun or DDA research. So with this library, we can go to these maps to extract the peak, and then we can score these peaks using open source, for example. So here, the retention time information can be used together with all the other parameters. And then this peptide can get identified and quantified, therefore also the proteins. So in an alternative way, you can try to analyze the real-time correlation between the MS1 signals and MS2 signals. So then you use the retention time already in the very beginning and extract the MS2 spectra. So this we call pseudo MS2 spectra. And these spectra, once extracted, can be used for data searching tools that are conventionally used for the shotgun proteomics. So these actually are 2 methods that already represent what I want to discuss in this lecture. So two concepts, the targeted data analysis and then the untargeted data analysis. So if you compare the left panel and the right panel, you can see some core features which are differentiated between these two. First, in the left, in the targeted data analysis, we need to rely on some spectral library. And normally, the retention time is used in the final step to perform the scoring for identification. So in the right, the retention time is used in the very beginning so that you can directly extract the spectrum. So this essentially differentiated to each other based on the step of information extraction. And this, I think, is a very important concept that we need to clarify in the very beginning. So then in the following slides, I will focus on each of them to explain how they work. So first, the targeted data analysis. If you think about the targeted data analysis, it can be done in small scale or large scale. It's been already covered some. And there will be some overlapping in slides, but that's actually on purpose so that we can link the talks between each other. So for the small scale, the smallest scale will be a peptide. Here you can see, again, this mixed spectrum from SWATH containing the fragment RNAs from any peptide in this precursor isolation window from 600 to 625. So this essentially contains the MS2 information for a lot of precursors. So that's why it's very complicated. However, our targeted peptides are actually hidden within this kind of MS2 signals. So with something that we know, this spectral library, which Ben talked about this morning, we can try to extract the RNA traces of all of these fragments over the LC gradient. So once these traces are overlapping to each other and you don't see a lot of noise here, this is generally a good indication of a confident peptide identified. So this is actually the step where I mean is the information extraction makes the targeted data analysis unique. So in this original paper of SWATH, we actually proposed this targeted data analysis strategy. And I want to show you here a more vivid plot. So where I used the peptide synthesis to synthesize this heavy isotopic standard. And then I will talk more about this in the biomarker application lecture tomorrow. But this is, we just say this is a peptide that's synthesized, which is heavy. And then I performed a dilution experiment from 100 femtomole to very low by threefold dilution. So this peptide is corresponding to a nectin-1 in human plasma proteome, which can be potentially a biomarker. So I can spike in this dilution curve into the background of the glycoprotein digest from 0.5 microliter of human plasma. So you can see if we perform the SWATH data analysis and then extract these traces based on the spectral library we gained from this heavy peptide, these peaks are very much the same to the SRM peak in Skyline. And you can essentially perform any analysis that you are familiar with Skyline to this kind of data set. So from here, we can say, okay, the intensity is decreasing along the dilution, and we can call a limit of detection here. So essentially, in this paper I published 3 years ago, we actually spiked about 40 heavy peptides, and then we tried to quantify the endogenous peptide concentration in human plasma. Interestingly, we quantified 37 peptides by SRM and 33 by SWATH. And if you look at the dynamic range, you can see SWATH and SRM are doing equally well. Maybe SRM is slightly better at the lower end. So nevertheless, the level 3 CV between the two methods are very close to each other. So I just want to point out this is a simple example of small-scale analysis where there is no FDR control. But if you do everything correctly, you can already achieve some data set that is publishable and meaningful. So during this course, we already learned two components that can allow us to move from the small scale to a big scale. So here these two components are first the data acquisition introduced by Ben, and also the assay library generation introduced by Ben. So here it is one component. Another one is the
I need the key for downstairs.
okay, so I think it's in my coat, so in the pocket. So then the mProfet was introduced by Ludwig, and we have the mProfet tutorial 7. That is, so you should be already very familiar with mProfet. So if you combine these two components, in theory you can already perform this automatic large-scale targeted data analysis of SWATH or DIA data. So I can skip these 2 slides, which essentially described the assay library as an input for data analysis. I just want to point out, so this is the format of the final SWATH library, and there are actually more information behind this library which you can rely on to perform the scoring. So for example, now you know the sequence of all the peptides, so you should already know all the theoretical MS2 accuracy, all the theoretical MS2 iron with their M/Z value for this peptide. And also, you should know the ideal isotopic envelope structure or pattern for this each fragments. So there are more information behind this table that can be used for the final scoring. So just to reinforce what I want to say, so here you have a very complicated SWATH data, and in each window you have a complex MS2 spectra for which you can use the primary knowledge of one peptide with the spectral library to perform the identification. So you zoom in, you see a Y5 iron with its good envelope pattern of the isotopic pattern, and then you can perform this kind of identification using some information of co-elution, peak shape similarity, the correlation with the assay towards the peptide or towards the spectral library, and finally perform this targeted data analysis. So then in the next slides, we can discuss OpenSWATH. So this is a large-scale targeted data extraction software developed by our group. So you can visit this website for more information or have a try there. So essentially, the developers of OpenSWATH need to first convert the file formats, like from this .raw file or .wave file to this MZML file. And for the library, as Ben suggested, we need to convert from this SPTXT file to the trauma template. So this is essential to make the OpenSWATH work in the very beginning. And then you can use the retention time information to do alignment. So here is just the purpose is just to focus on certain window of this LC gradient, so that you don't need to go to all the other parts of this LC gradient. For example, 5 minutes or 10 minutes. And then you perform this chromatogram extraction for all these iron traces and rely on this feature scoring by OpenSWATH to score these different peaks. So by the statistical analysis provided by mProfet, we can assign the second peak is actually the true peak, the true identification of this peptide. So this is a general overview of OpenSWATH workflow. So now we discussed the input as the data set, and then we should discuss this process with the perspective of these scores. So OpenSWATH works with similar scores to SRM, which are based on the chromatography. So here we have the retention time score, the correlation, the co-elution score, the cross-correlation shape score, intensity score, the Gaussian distribution. For example, this peak should be following the Gaussian distribution, the signal-to-noise score, and the relative iron intensity score compared to the library. So moreover, because we are doing this high-resolute MS2 scan, we also have the spectrum-based scores, such as this iron series, the mass accuracy of all the fragments, the isotopic distribution, and then interfering isotopic overlap scores. So you need to have both scores. Open source actually make the usage of both scores to perform the identification. If you go to Skyline, if you perform mProfet-based SRM analysis or open source or mProfet-based source analysis, actually there are different scores that are enabled automatically. So in the open source study, the developers need to establish a golden standard as a benchmark for the paper. For the software. So what they essentially do is that they try to find all the stable isotopic labeled peptide standard that they can find in our group, which turned out to be more than 400 peptides, and then they make this 10 times dilution curve, then spike all these peptide mixtures into the background of water, yeast, and human digest. And we measure everything in triplicates. And the second step to establish this golden standard dataset is really to perform an extensive manual analysis. So here you can see that they annotate comprehensively more than 30,000 chromatographies. And then here you can establish, okay, according to my manual analysis, which peaks are true or which peaks are false. So this is the golden standard. So based on this golden standard, you can then benchmark the output of open source software. So here you can see this is the pseudo ROC curve, the receptor operation curve, where the x-axis denotes the false positive rate, and the sensitivity essentially means once your manual analysis suggests it is a true peak, how often does open source also think it is a true peak? And you can see with a very small false discovery rate, open source can correctly identify more than close to 90% of the peptides. So this is a bit unfair for open source because the experiment itself is a dilution curve. So that means you can infer the identity of the peptides from the high end to the low end. So if the peptide is very low at the low end, you can still infer its identity by manual analysis simply because you know what it looks like in the high end. But despite of this fact, we still have the AUC more than 0.9, so suggests this result is very decent. So if you compare the FDR, the real FDR estimated by the manual analysis and the estimated FDR from open source, you can also see they are highly agreed to each other. So a further point I want to make here is that under very small FDR, open source can already detect all the peptides that can be detectable if they are in the data. So this is essentially a multiple sample data set. That means we have a very high data completeness, which is very useful for the large scale proteomic analysis. So how about quantification? Now we discussed the identification. For quantification, you can look at all these intensities of 400 peptides and then compare it to the highest concentration, and then plot this box plot indicating the quantitative result from manual analysis and open source analysis for this peptide spiked into the yeast background. So again, you can see they are almost the same between these two methods. So this actually essentially benchmark open source in identification perspective and in quantification perspective. So after discussion about the input, the method, now how about the output? So open source will report an output at a fragment intensity level for each sample. So here you see this is essentially the output table where you have the sample information here and then the assays, and then for each fragment in this assay you also have iron peak areas as the intensities. Importantly, you have the Q value, which is actually the M score from mProfet scoring for all of these peak groups in each sample. So if you have a very low M score, this means statistically it is very significant to say this peptide is there. So here you can see that for the first two samples, this peptide can be identified, and most likely you cannot identify this in the third sample because of the high M score. So the low M score will be transformed into a high D score, the discriminative score, and the high M score will be transformed to a low D score. So here you can perform the target decoy database searching strategy. So as you already know from the tutorial, you can see the distribution of the decoy hits are actually following this Gaussian distribution. However, for the target hits, it is a skewed distribution. So by plotting this kind of information, you can now start to move your D-score cutoff from the right to the left, and at a certain point you can stop, and because here you already have 1% of the decoy hits. So this is essentially called FDR 1%. So after setting this cutoff, then you can report everything that is in the right bar here. And here you can see after the identification, you already assigned one peak to one line in this table. So basically this is the output of the open source. Just want to comment that open source just solve the question tier the peptide group identification and quantification. So for further questions like protein inference or statistical analysis, you need to go to AFQ, MSStats, or MapDIA to really illustrate your biological story. So quickly to use this data set again of this interlabel study to present you how open source is efficient in at least in our hand. So here you can see this HEC293 digest distributed into 11 labs worldwide of 6 countries, and each lab used the standard source workflow, and then the data suggests that they all identified more than 4,500 proteins on average, and this is essentially overlapping to each other between the labs. So if you look at the accumulated proteins detected, if different labs detect different list of proteins, this curve will go very rapidly up. Now they are actually very flat, suggesting that each lab not only detects the same number of proteins, but these proteins are also quite identical between labs. And importantly, I want to point out here that the ACFDR now is controlled at 1%, and we are working very hard to control the protein level FDR to 1% here. So essentially, we proposed this new method, which is about Q-value scoring, where we only use the highest score of any peak group for the certain protein in any of these labs to represent this score of the proteins. And then we perform the same target decoy strategy. So this would be essentially similar to some methods that were used in shotgun proteomics. So with this strategy, we can control FDR, at least especially the protein FDR, in a better way. So I will show you in a minute what I mean here. So this is a final identification result. This is an update from what Rudy showed you, and from here you can see that in our lab, using routine cell lysis, normally we can get this number under very well established FDR controlling system at the protein level. And we hope this is finalized result. So besides open source, PeakView essentially actually doing the identical thing. So in this publication, you can see, for example, from this AP-masked sample, they can perform DDA and the SWATH, and the spectral library can be built from DDA. And then this library can be used to extract the peaks from the SWATH maps to perform the identification and the quantification. So this also suggests that the SWATH quantification is much more reproducible than the DDA analysis, because if you look at all the peptides with the CV less than 20%, you can find SWATH mass identified more peptides and proteins at this region. And they also benchmarked the SWATH and PeakView data to this iTrack quantitative data they got. And for the biological story, you can see both methods give you identical result. So just to summarize this part, the open source and targeted data analysis, we have input, which is SWATH data, the input of spectral library, and then we use this XIC-based data extraction and mProfet-based scoring system. This is the process, and the output is this fragment-level quantitative data. So is there any question or? Yes?
Just a short one. This pan-human library, it's, I assume, for trypsin. Are there any libraries for other enzymes?
Yes, so recently we are thinking about maybe include other enzymes, but the pan-human library is indeed after tryptic digestion. And the reason is certainly because this is a kind of routine practice in proteomics that this can be maximized with the usage if we just provide assays using trypsin digestion. Yeah. Okay, so after present you how open source works, I would like to discuss some problems of targeted data analysis. So if you think about it, the biggest problem actually is the need of the library itself, right? So as you said, you really need a library first. And also, you need to further worry about the coverage of the library, the quality of the library, and there are some detailed questions like how to use the MS1 information. Do you want to use it as an additional scoring system, or do you want to use it as a filtering system? And there are other problems, such like the PTM. So if you have a PTM or naked peptide for a certain peptide sequence, sometimes they fall into the same source window. So how can you differentiate which peak belongs to PTM, which peak belongs to naked peptide? So these are many open research questions here. So besides these questions that are easy to understand, I would discuss more about the questions related to the library size. So let's say if we have two lists, the first list is very short, the second list is quite long. So in theory, you can split this second list into different small lists and perform the open source identification separately. And then you control the FDR for each of these lists. But if you do that, the result will be different compared to the scenario where you use this list as the integrated list. So as Olga discussed, this is called this multi-comparison problem. And this is also kind of still open question, how to do this in the best way for your specific biological story. So another impact from the library size can be illustrated by this FDR distribution for the target hits and the decoy hits. So this D-score distribution. So this is related to the prevalence of the target peptides in your library. So if you start from a sample-specific library or a pan-human library, a big library, the situation will be different, as Ben already covered some this morning. You can see here the decoy distribution are actually similar between upper and the lower case for small library and for big library. However, the distribution of the target hits are actually different. So it's the skewed distribution here, but here you have a very big peak recapitulating this decoy distribution shape. So let's imagine if you do the FTR controlling, you will start from the right to the left, and at certain time point or at certain position you stop, and you call FDR 1%. But let's imagine if you lose the FDR criteria. Let's say now we want 2% FDR. So what you then you move towards the left again. But then here you can you only include some small number of targeted hits because many targeted peptides are already identified at this region. However, in this case, if you slightly lose the FDR criteria, you will include much more targeted hits because of the abundant distribution at this area. So this illustrates why you need to care about the library size, and this will certainly affect the protein FDR also, as I showed here. So this is the HeLa cell lysate. If you look at the SWATH data, you find that if you use the sample-specific library or the pan-human library, this generates different results. So the peptide iron FDR or the peptide FDR can be controlled very well by M-score in both cases. However, in the right, you can see that even if you use a very small M-score cutoff, the protein FDR is already inflated very quickly. So this suggests that once you have a very big library, you really need to care more about the protein level FDR. And here we propose that you can use Mayu, for example, or you can use this Q-score system that I described to you in the interlab study. So is there any question about these things? Again, yes, great.
I'm not sure that I catch it. So basically you are claiming it would be better to use the sample-specific library?
So it's also an open question, but for the sample-specific library, you have less concerns for sure, because it is identified directly by a shotgun analysis, and you know that most of the peptide in your library actually belongs to the true proteins in your sample. But for the pan-human library, if you perform it correctly and you have additional strategy at the protein level to control the FDR, you can also use it, and you will normally get more identifications. So yeah, as there are also some comments this morning, like if you want to generate the sample-specific library, you can have multiple injections, for example. Yeah, these are some practical concerns. Yeah. Good. So after the targeted data analysis, let's try to look at some untargeted data analysis. Let's go back to these slides again. Now we are focusing on the right panel. So the untargeted data analysis was originally, the idea was from the MSE in Waters machine. So MSE is a acquisition where you have MS1 scan and MS2 scan in tandem, and then you oscillate the collision energy to get this continuous acquisition of MS1 and MS2 scans. So in that case, all the MS1 scan and MS2 scan are multiplexed. So if you use the retention time information in the very beginning, now you are able to actually tell this co-elution pattern from these three peptides are actually different. So if you use the real-time correlation between the MS1 signal and the MS2 signal, you have the chance to actually deconvolute these three peaks with their corresponding MS2 signals. And this is essentially called a precursor fragment relationship. And by using this relationship, you can have this pseudo MS2 spectra for which you can use for DDA analysis. So essentially, the Alexei Levinatsky group last year published the same idea, however now applied in the SWATH dataset or DIA dataset. So this is a MS1 map, these are MS2 maps. So they have internal relationship because the retention time is the same, and then you can rely on this retention time to perform the real-time correlation analysis between MS1 signal and any of the MS2 signal. So they used some graph-based algorithm and finally come up with a lot of precursor fragment groups for which you can illustrate this pseudo MS2 spectra to do database search using the traditional shotgun proteomics. So here you can see that they have this kind of group data, and then they can perform untargeted identification. This is based on the traditional shotgun database search against a sequence, or you can also use the spectral library-based search. So this is called the spectrum-centric search. So then you have identification. So Alexei's lab also suggests that from this identification, you can build an internal spectral library. So essentially, it's an extension step that you can perform a targeted extraction. So here you have this library, then you go back to each sample with the MS2 maps, and then you can perform a secondary step of identification similar to open source. And here you will have a high coverage of quantification. And here you can also include some external spectral libraries. And this is essentially called a hybrid method, which I think is a very interesting idea. In the supplementary, you can see the comparison between the DIA result to open source and the DDA. So here you can see that it seems open source even detected less peptides and proteins, and we think it's certainly related to the library that they used. But nevertheless, the idea is to show that the DIA empire achieves comparable result to open source and the DDA, at least in their hand. So after the publication of a DIA empire, there are recently 2 corresponding papers from Nature Method presenting us 2 other alternative softwares, Group DIA and the MS Split DIA. So they are quite similar to DIA empire, but with some differences. This group DIA was actually developed by a Chinese group. Interestingly, they proposed that they can use additional information coming from multiple files. So here you can see, for example, there are 3 files, and you know that this peptide can be high in the first 2 files and low in the 3rd file. So MS1 tells you this relative quantification information. So ideally, if you have a correct MS2 peak corresponding to this MS1 quantitative information, then this MS2 peak should show you the same relative quantification result. So then you can really say, okay, only this purple MS2 signal belongs to this MS1 signal. So this is a kind of additional information that you can use from multiple samples to somehow refine this pseudo MS2 spectra you get from a source data set or DIA data set. So I think it's a very interesting point of view. If you look at the result, it is also conceivable that at a small number of data files, it seems the software is close to DIA empire. But if you have more data files, and it seems the group DIA gives you more identification. Of course, this is only from their lab. We need to verify it maybe in other labs, but I definitely think it's an interesting idea that people should try. And so for the MS Split DIA, it was developed by Neuro Bandaya group, where the MS Split actually is abbreviation of mixed spectrum partition using libraries of identified tandem MS spectra. So what does it mean? You have a mixed spectrum here, and at the very first step, they already use the spectral library to do the projection. So they can project this mixed file into the peptide entries in this library. So they used the library and the M/Z information in the very beginning to score this multiplexed spectrum by partitioning it into different entries. And then you can remove some independent matches. Sometimes the split is not good enough, so you can remove some 2 similar spectra, and then you can further use the retention time information here to perform the scoring. And if you use targeted decoy system, you can give a peptide level FDR. So this is their way of doing it. Interestingly, they compared different samples. You can see human sample is of highest complexity. So this kind of mixed spectrum can sometimes contain 10 peptides. So just additional comments. If you think about this strategy, it's very close to DIA empire, but also differs in the very beginning where they need a spectral library. So essentially, it's not a spectral library-free method. And it's also hard to say if it is a targeted method or untargeted method, because the first step is already based on the library. So that's why we think maybe this targeted and untargeted term should be revised to this peptide-centric or protein-centric term. So because, peptide-centric or spectrum-centric term, because here it is clearly a spectrum-centric method. So with this, I would go to the summary if you don't have further questions. Yes, please.
A quick question, if you go back a couple of slides back. Here, yeah, the previous one. The next one, sorry. Mm-hmm. Yes, here. Do you know what kind of gradients they were using here?
What kind of
gradients?
I think it's just a normal gradient. I have no idea what is. No,
the numbers are rather low in here.
What do you mean by the numbers?
It's peptides. It's not proteins here.
Number of peptides identified.
Yeah.
Yeah, but this is the yeast, right? And this is human. So I think essentially the data set quality is not the concern here. It's more about the software. Yeah, but if you developed your software on 5,000 peptides, then how do you know how it performs on 50,000 peptides? Yeah. So if you look at the original DIA IMPI, they also don't have a large number. So I think there's still some improvement about the data set, but it's not related to the software, I think. Yeah. Okay, so this brings me to the summary. First, I would introduce this terminology change. So this was a review from McCall's lab, where they present the idea of this spectrum-centric and peptide-centric. So open source will belong to this peptide-centric because you have a target peptide in your hand, and then you go back to the source maps to perform the scoring of certain peaks. And for spectrum-centric, you can start from both DDA result or DIA result. So you have a mixed spectrum here or a simple spectrum here. And after a traditional shotgun proteomic workflow, you can assign this one peptide, for example, normally to one spectrum in the DDA case, but multiple peptide identification to the mixed spectrum in the DIA case. So this is the terminology change. So finally, about these softwares, you can see that open source, PeakView, Skyline, or Spectronaut, these all belong to the idea of targeted data analysis, and it is essentially based on a spectral library. And recently, we prefer to call it peptide-centric. And for these softwares in the lower case, you can see PRGS, DIA Empire, MS Split, Group DIA. This belongs to the untargeted idea. And of course, sometimes you don't need a spectral library, but for MS Split, you need a spectral library. And essentially, we now call it spectrum-centric. Finally, I just want to present you unpublished data from a previous colleague, Pedro Nevado, and in their manuscript, they actually try to benchmark all these softwares towards some standard samples. So in A and B, you have two different samples with different fraction percentage from E. coli, yeast, or human, and by performing source analysis respectively for A and B, you can essentially compare the identification and also relative quantification between these two samples. So based on all of these different softwares, we can identify consistently more than 4,500 proteins. So interestingly, all the software also achieved a very similar level of quantification. So this is trying to tell you that no matter which kind of method you are using, if you perform it correctly, most likely you will end up with similar result. So this brings me to the summary. So I present you the concept difference between targeted and untargeted data analysis. So the input and output of open source analysis. And this XIC-based analysis combined with DIA facilitate very high level of reproducibility compared to the manual analysis. And then the targeted analysis, now we call it peptide-centric analysis, scales from SRM-like analysis up to a very global studies. And you need to pay some specific consideration to the protein FDR for the large libraries. And then the spectrum-centric method are being available. And of course, this hybrid method could be further used, and that could be interesting to watch in the future. And thank you for your attention. If you have any questions.